metabolism of amino acid or proteins. Same metabolism of amino acid or proteins. A individual adult has 100 gram of free amino acid which is known as a amino acid pool of your body. तो आपकी बॉडी में 100 ग्राम अमीनो एसिड्स रहेगा, उसको अपन बोलते हैं अमीनो एसिड पूल ऑफ योर बॉडी. ग्लूटामेट, ग्लूटामाइन, दे ओनली कंस्टिट्यूट 50% ऑफ जो 100 ग्राम अमीनो एसिड का पूल होता है उसमें का 50% क्या रहेगा भाई, ग्लूटामेट एंड ग्लूटामाइन. बिकॉज इन दोनों के ऊपर ही बहुत सारा चीज चलता है. बिकॉज इंडिविजुअल अमीनो एसिड विल बी मेटाबोलाइज्ड एंड it is converted into lastly glutamate or glutamine, because these are the intermediate in the, it is intermediate in the various metabolic reactions. Glutamate essential amino acid उसमें 10% रहेगा. 50% amino acid pool is composed of glutamate and glutamine and 10% you have essential amino acids. There is no storage of amino acid in case of, as there is no storage form of amino acid as in case of carbohydrate. Okay, जैसे होता है आपकी बॉडी के लिए ग्लूकोज लगता है, ग्लूकोज का कन्वर्जन होता है ग्लाइकोजन में, ग्लाइकोजन इज स्टोरेज फॉर्म ऑफ ग्लूकोज. सो देयर इज नो स्टोरेज फॉर्म ऑफ अमीनो एसिड इन द बॉडी, सो दे आर नॉट stored. So that's why you have amino acid pool of your body. So 100 gram free amino acids they are present in the body. So out of that 50% is composed of glutamine and glutamate and glutamine. 10% of amino acid pool is nothing but having essential amino acids and a PM vital. Then there is no storage form of amino acid. As in case of carbohydrate there is a glycogen, as in case of lipid there is a triglyceride. आप बोलेंगे proteins, protein they are the polymer of amino acid, they have their own function, they are not a pool of your, they are not a storage form of amino acid. Now 300 to normally 300 to 400 gram of protein per day is required, which is constantly metabolized, which is constantly degraded and synthesized, which represent the body's protein turnover. तो नॉर्मली 300 से लेके 400 ग्राम प्रोटीन पर डे आपको लगता है, या फिर आपकी बॉडी बनाती है या फिर उतना डिग्रेड करती है. देयर इज वन प्रोटीन नोन एज अ बायोक्वेटिन. ओके, देयर इज अ प्रोटीन नोन एज अ बायोक्वेटिन, यू साइलेंट बायोक्वेटिन, a small peptide which increases the protein degradation. A 30 to 50 gram protein must be supplemented per day in a diet to maintain a nitrogen balance of your body. तो इतना normally लगता है protein. Now metabolism of amino acid. There are mainly two reactions. Transamination reaction is there and deamination reaction is there. Now amino acids will undergo reaction like transamination followed by deamination to liberate to form a ammonia. So protein का भी metabolism में दो ही, there are two reactions only. So transamination followed by deamination, and if they are sequenced, ये दोनों में most of the time transamination होता है, transamination और trans के बाद में glutamic, in case of glutamic acid, glutamate, so there is a transamination followed by deamination. So sometimes it is known as trans deamination, sometimes it is known as trans deamination for liberation of ammonia. ग्लूटामेट का मेटाबॉलिज्म ओनली, ग्लूटामेट का मेटाबॉलिज्म दैट ऑकर्स ओनली इन द लीवर. बाकी ऑर्गन्स में बाकी सब का मेटाबॉलिज्म होता है, बट ग्लूटामेट का मेटाबॉलिज्म ओनली दैट ऑकर्स इन द लीवर. ग्लूटामेट का मेटाबॉलिज्म ऑफ ग्लूटामेट ट्रांसअमाइनेशन फॉलोड बाय डीअमाइनेशन दैट टेक्स प्लेस इन द लीवर ओनली. नाउ अमाइनो group of amino acid will combine with the ammonia to form a urea, which is the end product of your protein metabolism as well as amino acid metabolism. Amino group of amino acid will combine with the ammonia to form a urea, which is easily excreted from your body. So urea is the end product of your amino acid metabolism. Carbon skeleton remained after transamination, now which is converted to keto acid and which can be used, which can be utilized to generate the energy or to synthesize the glucose or the formation of ketone bodies or formation of non-essential amino acids. तो क्या होता है ट्रांसअमाइनेशन में कि ट्रांसफर ऑफ अमीनो ग्रुप टू द अमोनिया टू फॉर्म ए यूरिया. एक पॉसिबिलिटी ये होता है यूरिया विल बी एक्सक्रीटेड एंड व्हाट एवर कीटो एसिड इज जनरेटेड, है ना. जैसे ग्लूटामेट का अगर ट्रांसअमाइनेशन करेंगे तो है ना ग्लूटामिक एसिड बनेगा, है ना. तो ग्लूटामिक एसिड इज यूटिलाइज्ड फॉर द सिंथेसिस ऑफ न्यू अमीनो एसिड और सिंथेसिस ऑफ शुगर और उसका वो अलग अलग उसका मेटाबॉलिज्म होता है, सिंथेसिस ऑफ ग्लूकोज और एनर्जी टू प्रोड्यूस एनर्जी और टू प्रोड्यूस अ कीटोन बॉडी और टू प्रोड्यूस अ नॉन एसेंशियल अमीनो एसिड. ये हो गया. ओके, नाउ simultaneously there will be transamination, transfer of amino group from one one amino acid to the another keto acid to form a new amino acid and new keto acid. ऐसा भी हो सकता है, we have already seen. So, now transamination means what? Transfer of amino group. Transamination means transfer of amino group from one, transfer of one amino group from amino acid to the keto acid by enzyme known as a transaminase. Now transaminase is dependent on pyridoxal pyrophosphate as a coenzyme. जो pyridoxal pyrophosphate होता है विटामिन B6 का, so it is a coenzyme required for transaminase. Now this process is a reversible process. Transamination reaction is important for production of non-essential amino acids. Okay, then production of energy, then production of, okay. Now glutamate is the only amino acid which will undergo oxidative deamination. Oxidative deamination. एक theory भी है उनका, theory बोलते हैं ping pong bye bye. There is one theory, ping pong bye bye. Theory is proposed by Snell and Brustine for the transamination reaction. Theory याद रखना है, ping pong bye bye. कुछ भी नाम देते हैं, ping pong bye bye. You can check the theory, ये नाम वही है, बस यहाँ रेड में लिखा है कि ये BYE है या फिर BI है, बस स्पेलिंग मिस्टेक के लिए पिंग पोंग बाय बाय प्रोनाउंसिएशन सेम ही रहेगा. ये हो गया ट्रांसअमाइनेशन. नाउ deamination means removal of amino group from amino acid to form ammonia or urea. Deamination मतलब क्या है? रिमूवल ऑफ अमीनो ग्रुप. ट्रांसअमाइनेशन मतलब क्या है? ट्रांसफर ऑफ अमीनो ग्रुप. उसके नाम में ही रखा है, ट्रांसअमाइनेशन मींस ट्रांसफर ऑफ अमीनो ग्रुप, deamination means removal of amino group. Now oxidative deamination catalyzed by glutamate dehydrogenase. So oxidative deamination that takes place in the liver only. Oxidative deamination that is done by enzyme, or that is done by enzyme known as glutamate dehydrogenase. Now this glutamate dehydrogenase activity is present in the liver only, and that's why liver में ही oxidative deamination होता है. Oxidative deamination by amino acid oxidase, and that mostly occurs in the liver and kidney. Are you getting or not? Metabolism of amino acids. दो रिएक्शन मोस्टली होते हैं एंड इंडिविजुअल अमीनो एसिड का मेटाबॉलिज्म अलग अलग होता है. am I audible to you? Am I audible to you? Non-oxidative deamination to generate ammonia without undergoing the oxidation. Now it is done by amino acid dehydratase enzyme to form a respective alpha keto acids. Second होता है amino acid desulfide. Okay, amino acid desulfidase. There are two enzymes responsible for the non-oxidative deamination. First होता है amino acid hydratase, okay, hydrase, and second होता है amino acid desulfidase to produce a respective keto acid and liberate the ammonia. The ammonia liberated in the transamination and deamination is used or is utilized for the production of non-essential amino acids, then purine bases, pyrimidine bases, then amino sugar. The extra ammonia is removed in the form of urea. Now, disposal of ammonia. जो ammonia, so whatever ammonia is formed in the metabolism of amino acids, so that ammonia is toxic to your body. Ammonia क्या है? Toxic होता है. Ammonia is toxic to your body. So it will cause sedation, glaucoma, and all that. Lot of problems are there with ammonia. Now, disposal of ammonia. So ammonia is disposed of, it is removed from your body either in the form of as it is. Some animals, they dispose ammonia, they excrete ammonia. So based upon that, okay, so for example aquatic animals, fish, etc., so they dispose of ammonia in the surrounding water. So ammonia as it is जो बना है वो remove करेंगे. So that's why they are known as aminotelic. So the animals which removes ammonia as it is in the surrounding known as aminotelic. In the few animals, for example reptiles and birds, उसमें क्या है कि ammonia is converted into uric acid, and then uric acid is excreted. That's why they are known as uricotelic. So ammonia is excreted as it is known as aminotelic. So example in the aquatic animals. Uricotilic means ammonia is converted into uric acid and then it is excreted. In case of reptiles and birds, so it is known as a uricotilic. Now ureotelic means what? So ammonia is converted to urea in the mammalians. Then as I say humans में जानवरों में तो ammonia urea में convert किया जाता है and urea is soluble in the water and it is excreted in the urine. So humans are ureotelic. So based upon that है ना अमोनिया का डिस्पोजल कैसे करते हैं कुछ कुछ एनिमल्स अमोनिया एज इट इज रिमूव करते हैं कौन एक्वाटिक एनिमल्स जैसे फिश वगैरह उनको बोलते हैं अपन अमाइनोटेलिक ammonia is converted to uric acid and then it is excreted in case of reptiles and birds and that's why they are known as a uricotelic. The ammonia is first converted to urea and then it is excreted in the mammalians, this is humans. So we are known as a ureotelic. तो ये आपको 12th में हो चुका होगा अमाइनोटेलिक यूरियोटेलिक यूरियोटेलिक. Now toxicity of ammonia. If ammonia is accumulated in your body, so increase in the level of ammonia is harmful to the brain. Ammonia when it accumulates in the body, so there result in the slurring of speech. आपका speech clear नहीं आएगा. Slurring of speech है ना एक दो पैक लगाने के बाद में जैसे बोलता है आदमी वैसे बात करेगा. Slurring of speech, blurring of vision. It can cause tremors. It may leads to coma and finally death. So urea is a very dangerous. So that's why liver failure है ना kidney failure in both cases there is accumulation of urea in the body and that's why patient may go in the coma. यहाँ पे MAY may go into the coma and may cause death. तो वैसे ही जो ज़्यादा बेवड़ा वाला होता है ना तो जिसका लीवर खराब होता है तो उसको वो मरता क्यों है मरने का कारण वही होता है कि उसके बॉडी में अमोनिया एक्यूमुलेट होता है अमोनिया उसमें अमोनिया जनरेट होता है बहुत सारा एंड लीवर खराब होने के कारण यूरिया साइकिल नहीं होता अमोनिया यूरिया साइकिल बंद होता है और अमोनिया और ज़्यादा एक्यूमुलेट होता है वो कोमा में जाता है. Now urea cycle is known as a also known as a urea cycle is known as a Krebs-Henselt cycle. Krebs-Henselt cycle or also known as a ornithine cycle. Urea cycle is also known as a Krebs-Henselt cycle also known as a ornithine cycle. इसको और एक नाम है ornithine cycle. So it was the urea cycle was first metabolic cycle which was elucidated by Hans Krebs and Kurt Henselt. Hans Krebs and Kurt Henselt. That's why it is known as a Krebs-Henselt cycle. Urea is synthesized in the liver. Liver में बनता है urea है ना and transported to a kidney for excretion. Now the urea has a two amino group. Urea का structure क्या होता है दो amino group NH2C double bond O NH2. Now these two amino groups comes from one amino group okay two amino group from NH3 and other from aspartate. तो एक अमोनिया आता है और दूसरा अमोनिया आता है aspartate से. So urea cycle is a 5-step reaction. First two enzymes for urea cycle they are present in the mitochondria and all other enzymes are there present in the cytosol. So urea cycle takes place in the mitochondria as well as cytosol. दोनों में. So first step is nothing but the synthesis of carbamyl phosphate CPS in the and it is the irreversible step and rate limiting step in the urea cycle. 4 ATPs are consumed over here. 4 ATPs are consumed over here in the urea cycle. See the urea cycle. तो ये ट्रांसअमाइनेशन डीअमाइनेशन रिएक्शन ऑलरेडी कैसे होता है एक ट्रांसफर ऑफ अमीनो ग्रुप टू अनदर कीटो एसिड ये कीटो एसिड है उसको ट्रांसफर करेगा उससे बनेगा नया अमीनो एसिड और इसका बनेगा नया कीटो एसिड known as a transamination. When see transamination reaction again, so this is a glutamate. ये क्या है glutamate मतलब glutamic acid है. So इसका अमोनिया लेके so there is transfer of ammonia from glutamic acid to the pyruvate. This is structure of pyruvate. You can see. I think am I audible to you? Audible आएगा. This is a glutamate and this is a pyruvate. So what is pyruvate? It is a keto acid. ये क्या है? Keto acid है. Keto acid. So ammonia or amino group that is taken and it is transferred to a pyruvate to form a alanine. That is a new amino acid. And whatever skeleton is remained, this is now it is known as a alpha ketoglutarate. it will enter into a Krebs cycle. Okay, now it is catalyzed by enzyme known as alanine amino transferase ALT amino transferase, and it requires a pyridoxal pyrophosphate as a coenzyme. This is a reversible step known as a transamination reaction. Now deamination reaction क्या होता है? So deamination means removal of ammonia, removal of amino group from the amino acid. Deamination, okay, that occurs in the liver. So transamination and transamination or deamination, these are the two reactions. Transamination से क्या होता है? Amino acid is converted into corresponding keto acid by transamination. For example, alpha ketoglutarate. Okay, and this is converted into any amino acid is there, doesn't matter. So transamination हो सकता है, deamination हो सकता है in the tissues, and finally it will become alpha ketoglutarate, glutamic acid. Finally glutamic acid का deamination हो गया, alpha ketoglutarate बनेगा and glutamate in all tissue. But now for the deamination of glutamate, enzyme required for the deamination of glutamate, that is a glutamate dehydrogenase enzyme. So that is available in the liver only, and that's why it occurs in the liver. So बाकी सब deamination, transamination बाकी amino acid का हो सकता है आपके बाकी all tissue में, in the all tissues. Glutamate का deamination होगा आपका liver में, right? Because there you require an enzyme known as glutamate dehydrogenase, which causes deamination of glutamate. Now deamination means what? Removal of this ammonia. ये ammonia जो remove होगा, you can see over here, removal of ammonia. It is nothing but the alpha ketoglutarate. And NADH, you know that NADH will give you 3 molecules of this ammonia is combined with the another aspartic acid molecule. Aspartic acid, उसमें दो ammonia group होता है, दो amine group होते हैं. One ammonia and of the aspartic acid and this ammonia will combine together to form a urea. Okay, and that is known as urea cycle. That is known as urea cycle. So these are the two reactions: transamination and deamination. Now what is urea cycle? You can see over here. So glutamate or glutamic acid is there. So there is a removal of ammonia from this glutamic acid by deamination in the liver. This ammonia. Now this ammonia can be utilized for different purposes. Ammonia can be utilized for the synthesis of amino sugar. This ammonia group can be transferred to another keto acid to form new amino acid, non-essential amino acids. And this ammonia can be used for the synthesis of pyrimidine, purine bases. After deamination of this glutamate, you will get an alpha ketoglutarate. Okay, after deamination, it is done by carbamyl. Okay, now this occurs in the liver in a cytosol. Carbamyl phosphate synthase 1. This enzyme is very important. Carbamyl phosphate synthase 1. What it does? ये जो ammonia है, it will convert this ammonia with the by taking this ATP. It will convert into it will combine. So it will convert into carbamyl phosphate. Carbamyl phosphate. Carbamyl phosphate synthase 1 will convert ammonia, free ammonia into carbamyl phosphate. Now this carbamyl phosphate, okay, now it enters into urea cycle. Now what happens? So ornithine amino acid is there. Ornithine amino acid is a non-essential amino acid. This ornithine amino acid will combine with this carbamyl phosphate to form a citrulline by ornithine transcarboxylase enzyme. Ornithine transcarboxylase. So citrulline will be there. Okay, तो यहाँ से जो है ना phosphate ammonia वो सब यहाँ पे citrulline बन गया. Citrulline is converted into arginosuccinic acid और arginosuccinate. citrulline के साथ में citrulline के साथ में oxalic acid oxalate को भी combine हुआ by the enzyme arginosuccinate synthase. It will convert into arginosuccinate. यहाँ पे देखो, यहाँ पे एक एक्स्ट्रा अमोनिया है ऑर्निथिन में, है ना, एक्स्ट्रा अमोनिया. तो यहाँ पे जो अमीनो ग्रुप है और यहाँ पे वो कार्बोमाइल फॉस्फेट एंड ऑर्निथिन कंबाइन होकर ये स्ट्रक्चर बना है NH2C+O सिट्रुलिन. उसमें एक ऐड होता है, कौन सा, ऑक्सलेट ऐड होता है, बनता है आपका आर्जिनोसक्सिनेट. अर्जिनोसक्सिनेट। उसके बाद में अर्जिनोसक्सिनेट से it will be converted to arginine by arginosuccinase amino acid. Arginosuccinase enzyme will convert arginosuccinate into arginine with the loss of fumaric acid. The loss of fumaric acid. Fumaric acid. यहाँ पे citrulline में oxalic acid add होता है fumarate and there is conversion of this arginosuccinate to arginine. Now arginine is nothing but the arginine is a basic amino acid है ना। तो यहाँ पे देखो NH2 C double bond NH2 NH2 arginine. Now from this there is a liberation of urea over here and arginine is converted to ornithine by arginase amino acid arginase enzyme. So arginase enzyme convert arginine into ornithine with the liberation of urea. And now this ornithine will again take part in the another molecule of carbonyl phosphate. That's how urea cycle is there. So are you getting urea cycle or not? Urea cycle समझ में आ रहा है? Should I explain again? Urea cycle again explain करता हूँ मैं. Am I audible to you? Audible आएगा? So you can see over here glutamic acid और glutamate. ठीक है, glutamate से अमोनिया है ना निकाला जाएगा. कहाँ कैसे निकाला जाएगा? Deamination of glutamic or glutamic acid result in the removal of ammonia and formation of alpha ketoglutarate. ये मॉलिक्यूल क्या करेगा? It will enter into the Krebs cycle. ठीक है, this ammonia with NH4, so it is taken up and conjugated with the phosphoric acid, okay, by the enzyme known as a carbamyl phosphate synthase 1 CPS, carbamyl phosphate synthase. So there is synthesis of carbamyl phosphate. ये इसको बोलेगा पे carbamyl phosphate. ये अमोनिया क्या होगा? ट्रैप होगा इसमें. Okay, now it will enter into the Krebs cycle. It will enter into the urea cycle. How? The urea cycle start with the conversion of condensation of ये ornithine amino acid होता है एक. It is a non-essential amino acid. So it is only found in the intermediate in the urea cycle. So ornithine will combine with the carbamyl phosphate to form a citrulline by the enzyme known as a कौन करेगा? कार्बामोइल ट्रांसकार्बोक्सिलेज. देखो सब यहाँ पे ही चलेगा कार्यक्रम इतने एरिया में. देखो NH3 प्लस था उसमें ये फॉस्फेट वगैरह ऐड होके NH2C डबल बॉन्ड ONH ये स्ट्रक्चर बनेगा. और दिस इज नोन एज सिट्रुलिन. और सिट्रुलिन में ये ऐड होगा क्या? और एक अमीनो एसिड ऐड होगा, एस्पार्टिक एसिड. यहाँ पे एस्पार्टिक एसिड, तो एस्पार्टिक एसिड ऐड होगा. सिट्रुलिन इज कन्वर्टेड इनटू arginosuccinate by the addition of this aspartic acid by enzyme arginosuccinate synthase. और this arginosuccinate ये स्ट्रक्चर आ गया यहाँ पे. यहाँ से एक अमीनो एसिड भी निकलेगा, ये अमीनो एसिड निकलेगा, यहाँ से एलानिन निकलेगा. Now arginosuccinate is converted into arginine. Arginosuccinate is converted into arginine by enzyme arginosuccinase with the release of fumaric acid. With the release of fumaric acid ये रिलीज हो जाएगा. CO minus CH2 CH2 बॉन्ड CO minus. बचेगा क्या? अर्जिनिन बचेगा. और अर्जिनिन तो बचा ये उतना. This is arginine. Arginine is converted into ornithine by enzyme arginase with the release of urea. NH2C double bond O NH2. Now ornithine will take part again in the urea cycle. This is known as the urea cycle that occurs in the liver. First two enzyme for urea cycle. Okay. First ये जो है that occurs in the cytosol and this occurs in the mitochondria of hepatic cell. Is that clear or not? Okay, then metabolic defect of urea cycle. Urea cycle में अगर defect हो गया तो क्या होगा? कार्यक्रम हो जाएगा आपका metabolic defect in the urea cycle. So first one is known as hyperammonemia type 1. Hyperammonemia type 1. हाइपर मतलब ज़्यादा, अमोनिया में अमोनिया मतलब अमोनिया, मिया मतलब ब्लड में अमोनिया बढ़ जाएगा. हाइपर अमोनीमिया टाइप फर्स्ट इज ड्यू टू द डिफेक्ट इन द carbamyl phosphate synthase first. So if your ammonia is not getting trapped, अगर अमोनिया जो बन रहा है वो अमोनिया अगर कन्वर्ट नहीं हुआ कार्बामाइल फॉस्फेट में तो यूरिया साइकिल नहीं चलेगा, है ना. तो डिफेक्टिव एंजाइम इज देयर कार्बामाइल फॉस्फेट सिंथेसेस. Now, hyperammonemia type second. Hyperammonemia type second is because of the ornithine transcarboxylase. Ornithine transcarboxylase है ना. दूसरा जो है वो अगर डिफेक्ट है तो hyperammonemia type 2. तीसरा होगा citrullinemia. तो यहाँ पे citrullinemia मतलब क्या, इंक्रीज इन द ब्लड citrulline लेवल. क्यों होता है, बिकॉज देयर इज अ डिफेक्ट इन द arginine सिंथेसिस. arginine सिंथेसिस नहीं होगा तो citrulline बनता रहेगा, तो citrulline लेवल बढ़ जाएगा, citrullinemia हो जाएगा. नेक्स्ट होता है आपका, नेक्स्ट डिफेक्ट होता है arginosacchinic aciduria. तो arginosacchinic aciduria मतलब क्या, तो arginosacchinic acid आपके बॉडी में बढ़ जाएगा और यूरिन के थ्रू निकलेगा, तो arginosacchinic aciduria बोलते हैं उसको. That is due to the defect in the arginose enzyme. लास्ट में arginase enzyme अगर नहीं है arginase, तो हाइपर arginemia होगा, arginine बढ़ जाएगा बॉडी में. These are the metabolic defect of urea cycle. ये याद रखना है आपको, कभी कभी क्वेश्चन आता है इसके ऊपर. Now metabolic defect of individual enzymes, sorry individual amino acid. तो ये बहुत ही ज्यादा इंपॉर्टेंट है, इसके ऊपर बहुत बार क्वेश्चन आ चुके हैं, बहुत बार क्वेश्चन आ चुके हैं. Okay, so metabolism of individual amino acid we will see in upcoming sessions. थोड़ा शॉर्ट में आपको वो भी बोलूंगा मैं आपको अपकमिंग सेशन में, बट ये इंपॉर्टेंट है, पर ये लेते मेटाबॉलिक डिफेक्ट का मेटाबॉलिज्म ऑफ इंडिविजुअल अमीनो एसिड एंड देयर डिफेक्ट. नाउ glycine amino acid. So if there is a glycine amino acid metabolism defect in the metabolic enzymes required for the glycine or inborn errors है ना, inborn errors. बचपन से ही आपको मेटाबॉलिज्म में दिक्कत है. If there is a defect in the glycine metabolism, you will suffer from glycinuria. Glycinuria मतलब क्या, यूरिन. ग्लाइसिन मतलब ग्लाइसिन यूरिन में आ गया आपका, तो उसको बोलेंगे ग्लाइसिनुरिया. This is due to the defective renal reabsorption. Defective renal reabsorption मतलब क्या, कि नॉर्मली ये रीएब्सॉर्ब होना चाहिए ग्लाइसिन यूरिन में से, ठीक है, बट नहीं हो रहा है. मतलब क्या हो रहा है, यूरिन में आ रहा है. So that's why defective renal reabsorption that increase the serum level of glycine. Primary hyperoxaluria. Oxalosis बताते हैं, oxalosis. Defect, there is defect in the glycine transaminase. ग्लाइसिन को मेटाबोलाइज करने वाला एंजाइम होता है ग्लाइसिन ट्रांसअमाइनेज. अगर उसका डिफेक्ट हो गया तो क्या होगा आपका, there is an increased glycine level in the body. Increased urinary oxalate. Okay, oxalic acid यूरिन में ज्यादा रहेगा, and because of that also result in the oxalate stone. Oxalic acid का stones भी है, kidney stone. It is now known as a primary hyperoxaluria. Mainly it is due to the decreased protein tolerance. Defect in the glycine metabolism, glycinuria and primary hyperoxaluria. तो याद रखना है डिफेक्ट किसका है, ग्लाइसिन का है. नाम क्या है, ऑक्सैलुरिया. डिफेक्ट किसका है, ग्लाइसिन का है. इंपॉर्टेंट होता है. नेक्स्ट देखो, नेक्स्ट बहुत ही इंपॉर्टेंट क्वेश्चन इसके ऊपर आ चुके हैं बहुत बार. डिफेक्ट इन द मेटाबॉलिज्म ऑफ फिनाइल अलानाइन, so known as a phenylketonuria. PKU. The name is due to the metabolite phenyl pyruvate, which is a keto acid. तो क्या होता है, इसका डिफेक्टिव मेटाबॉलिज्म होता है तो फिनाइल पायरुवेट कीटो एसिड बनता है, and that's why it is known as phenylketonuria. So what is the defect? Defect in the phenylalanine hydroxylase. ये phenylalanine का मेटाबॉलिज्म करने वाला एंजाइम होता है, phenylalanine hydroxylase. So blocking of conversion of phenylalanine to the tyrosine. So there is a blockage conversion of phenylalanine to tyrosine. That's why phenylketonuria is there. Right? क्वेश्चन आता है इसके ऊपर, ऑलरेडी पूछा है एक बार. What are the symptoms of phenylketonuria? So mental retardation, defect in the myelin formation of nerve, insufficient synthesis of serotonin. Guthrie test is used to diagnose the phenylketonuria. Guthrie test, इसके ऊपर भी क्वेश्चन आ चुका है. So Bacillus subtilis is used as the organism for assay of phenylalanine. End product of your pyruvate, end product is into the phenyl pyruvate, which is a keto acid. So it can be detected by using ferric chloride test. This is phenylketonuria. Next होता है बहुत सारा का tyrosinemia 1, 2, 3 बहुत सारा है. So I think I will take it in the next session. So we'll see the remaining point tomorrow. I hope तुम्हाला